For many years we have been using DNA for species identification and other advanced studies. Recently we have this term we call eDNA, environmental DNA, which is DNA mixed from all kinds of species in the environment. And in the recent years there has been a lot of interesting studies. One of the problems, however, has been that if you want to answer questions of how many species are there in this sample, we have been challenged by a lot of, you could say, analytical errors during the sequencing and the PCR, and our algorithms for removing those errors have simply not been good enough. We were relatively annoyed with this fact, so we put a lot of effort into trying to see if we could remove further these errors to get more precise ideas about the richness, species richness of eDNA samples. So our new study is about a computer algorithm that we developed to, you could say, to clean up in dirty data, so you could get reliable biodiversity estimates. So basically you can get a much better picture of how many species are present in a sample with mixed DNA. The strong thing about this study is that it's been done as part of a project we call Biowide, where we have sampled the biodiversity in 130 different locations in Denmark. And what we realized was that we had these error patterns were very reproducible between our sites. So what the algorithm does is it takes information from the whole study and compares, so to speak, the abundance and occurrence of species and flags some of them as errors. What we show now is that if we apply this algorithm to our data, we get much more reliable estimates. So we postulate that this method will give us much better ideas about the richness of species in groups where we know very little.